Yes, so work group 5 was related to DNA repair, and then so especially then using the Comet assay to assess DNA repair. What I want to talk about today is the overview of the tasks and achievements of work group 5, an overview of the data analysis. So we collaborated with work group 1 on collecting DNA data on DNA repair in the biobank or in the data bank, and we analyzed those data. And I want to show some results, and then of course work that is still ongoing and future plans. So we had several tasks. So the overall objective was to develop a Comet-based DNA repair assay as a biomonitoring tool. So there were already a lot of assays available for measuring DNA repair, but we wanted to make it more specific for biomonitoring. And that means that we had to analyze the DNA repair data from the database. So I will show you today data of the base excision repair assay and the nucleotide excision repair assay. We also had to do some validation tests, and we did several small, not big validation tests, but just small experiments. And I will give you some examples of those of the results of those experiments. We had to come up with a framework for using the Comet assay as a DNA repair measurement tool in biomonitoring. And we published a review, and I will show that in the next slide. The protocol, we had to come up with a protocol for using DNA repair in human biomonitoring. And actually now the protocol has been accepted for publication, and we hopefully get the page proofs very soon. And then of course a training course on DNA repair, and we are training in DNA repair using the Comet assay in DNA repair, and we had a training course and several TSMs, and I will come back to those also later on. So with regard to that paper on making a framework to use DNA repair as a human biomonitoring tool, we have a paper in Mutation Research Reviews on DNA repair as human biomonitoring, which we go into aspects like use of surrogate tissue, because usually you would like to measure DNA repair in lung or liver or any tissue in which cancer occurs and not in white blood cells. But you have to use in biomonitoring samples that are easily accessible, which includes white blood cells. And we looked into the correlation between white blood cells and internal organs, the inter-individual variation, intra-individual variation, etc. So that can all be found in this paper. So we also from this paper we also identified several items that we still lack knowledge about, and one of those things, for instance, is that for measuring nucleotide excision repair, we use substrate cells that are either damaged by UV light or by BPDE, benzopyrene diol epoxide. And it's still not known, both work somehow, but it's still not known whether they give the same result. So we did a very small study in which we compared both assays, right? The UV-induced nucleotide excision repair shown in blue here on the left, and the BPDE as a substrate in the nucleotide excision repair assay shown in green. What you see is that there is a correlation between both. It's a small study, so it's of course you cannot say a lot, but you do see that there is quite some variation. Sometimes you see high repair in one assay, whereas then it is less efficient in the other assay, whereas you are using the same cellular extract, right? So the same DNA repair enzymes somehow may recognize the UV-induced damage differently from the BPDE-induced DNA damage. And of course, this can be assay variation, right? Because the amount of damage may be different, the type of damage is different. But this may also be real biological variation, that UV-induced damage is indeed differently recognized than BPDE-induced DNA damage. So I think we should look into that in more detail, and that is something that is, let's say, something that we should do in the future. Another item, if you want to go into biomonitoring, is that that the scoring is a bottleneck, right? We have now systems in which we can go with high throughput Comet assays. We can analyze images. High throughput is also automated, but then the scoring, that is sometimes very difficult. So you have to go through all those samples, and that takes a lot of time. So what we did is we compared several Comet assay scoring methods, which are either you can buy them like Comet 3 and Comet 4 from Perceptive Instruments, but we also use some freeware like Open Comet in IMAGE-J, the Comet Score 2.0, and COSPLOP. And the question was, do these systems perform equally well? So the different systems that we compared, they can either sometimes they are just, you have to score with their own settings, like in IMAGE-J. You cannot change the settings, but on the other hand, it can go into automated scoring, which makes it high throughput. These are the systems that we compared, and they can either yes or no adapt the settings, or they can yes or no be used for automated scoring. What we did is we took pictures of comets with varying types of damage, and then we analyzed those with all 5 programs. And most of the time we saw, when we make correlations between the 2 assays, which is shown on the left here, we saw that the percentage tail DNA with one scoring method be exactly the same as the percentage tail DNA with the other scoring method, right? So R squares were usually higher than 0.95 with a p-value smaller than 0.05. And also very important is that the slope was often very close to 1, which means that they both give the same value. There was one exception, and that was with IMAGE-J. IMAGE-J is actually having problems with DNA damage, samples with low DNA damage. So it does not recognize the damage often, and then it skips the cells for measuring the damage. Another way to look at that is with blend-Entman plots. What you actually want to see is a picture like on the right, where you see a horizontal relationship. So that would be ideal. But indeed, if you look into IMAGE-J, which is freeware and therefore people might use it, but we clearly see that there is a systematic error there when you compare it to other systems, right? So all packages seem to work really well, except then that Open Comet in IMAGE-J and also Metafair, but I did not show those data, seem to have some issues with low levels of DNA damage. At high levels of DNA damage, we see that sometimes you have to choose the center of the head, right? So that you can that the system knows, okay, this is the head of the comet, and this is not always easy to find, especially if you have high damaged cells. I think overall, high throughput scoring needs further attention. So then we did an analysis of the DNA repair database. Step 1 was gathering the data, and then thanks to Workgroup 1, this has been done, and we not only collected data on DNA damage, but also on DNA repair, and both nucleotide excision repair as well as base excision repair. So what we end up with is, in the hCOMET database, is that we have approximately 1900 data points of data from individuals, 1900 individuals on base excision repair, all using the raw compound and light to produce 8-oxo-DG as template. So there we had a nice database that we could link the repair to additional data such as gender, body mass index, age, DNA damage with the FPG Comet assay. The nucleotide excision repair database is much smaller, unfortunately. We have there approximately 600 samples, and it's also much more heterogeneous because some of them are related to V repair assay, others with the BPDE repair assay. Some have also a challenge assay, so you expose the cells first, and then you look at how the damage is removed in time. So therefore, this is much more difficult to analyze, and the only additional data point that we had in sufficient numbers is gender, right? So we analyzed the repair activity to all these additional data. But before we can start pooling the data, we need to standardize the data. And what we actually did is we take every value in a certain study and divide that by the mean of that study. And what you then get is an overall average of 1, but the coefficient of variation, which is the standard deviation divided by the average, remains the same for every study, right? But so 1 would be the overall value that you will find in each study, and also the overall pooled data. So then we looked into the effect of gender, and actually gender does have an effect on both base excision repair as well as nucleotide excision repair. And the difference between males and females is actually a little bit higher for nucleotide excision repair as compared to base excision repair. And what we see is that females usually score higher or have a better repair activity or higher repair activity as compared to males. So this again proves that females are the stronger sex among us. Then we also found an effect of body mass index, and there we made classes related to the different classes of body mass index. So smaller than 20, 20 to 24, 25 to 29, 30 to 34, and we also have highly obese subjects. And what you actually see is that when you are slightly overweight, that you have some kind of compensation by your DNA repair system. But then if you become obese, so let's say a body mass index higher than 35, then which is highly obese, of course, and then you see a strong decline in DNA repair activity. And this is interesting, we think, because also animal models for instance, the OGG1 knockout mice and NEAL1 knockout mice, they also show signs of obesity. So it's not known whether high obesity leads to deficiencies in DNA repair or the other way around. Then, of course, the overall idea is that a higher base excision repair activity should lead to lower damage. And we actually see that happening because if we group the people according to, let's say, a base excision repair activity lower than medium and a base excision repair higher than the medium in the group, and we relate that then to the FPG Comet assay, so not the normal Comet assay, because that's just strand breaks. The FPG Comet assay is specifically detecting similar lesions as compared to that are repaired by the base excision repair pathway, and we indeed see that those people with more repair have less DNA damage. So the conclusions from the database would be that base excision repair activity is related to gender, with females having a higher repair than males. We show that there is an effect of body mass index, and what I did not show, but we found no effect of age or smoking. The data on nucleotide excision repair is still, we are still investigating how we can work those out. So this is still under investigation. So to follow from that. And higher DNA repair was related to less DNA damage, which I think is also good to see, and is also expected. So what's now ongoing is that we are writing manuscript. We have a first draft almost ready, and then that will be distributed to the authors, to co-authors, for their comments. One of our deliverables was also to come up with a protocol. As I said, we now have a Nature Protocols paper accepted for publication. And in this paper, we have protocols for base excision repair. We have a protocol for nucleotide excision repair. We show how extracts need to be made. We have examples on how to calculate repair activity. There are also problem shooting chapters, so that if you have weird results, what is then the potential source of that result. And we also made a film to make things more visual, because we humans, we are visually orientated. I think if you show it, it's much clearer than a large piece of text. And these videos will, by the way, also be linked. There will be links in the Nature Protocols paper. Our last task was training. So we did a training course on the DNA repair assay in Norway in 2017, so already 3 years ago. And I think that was quite successful. And we had several STSMs. I'm not going to list them all, but we had learning actually the repair assay, and actually some of these people have already published papers on that assay. So what's next? How do we proceed? I think we still have to finalize the manuscript on the base excision repair database. We also have to analyze the nucleotide excision repair database, and we have to write the manuscript. And I think also when I relate it to Workgroup 7, planning the future, I think we can apply for funding. And I think that repair and obesity, we found that relationship between base excision repair and obesity. In my view, that would be a very nice subject to go forward. So that was Workgroup 5. I would like to thank everybody who participated in this work group. I don't have a list of all the people that were involved, but I want to thank everybody, and I think I will also thank you on behalf of Sabine, who was my deputy, and she did also help me out quite a lot. So thank you very much.
Thank you, Roger, and I'm sure there are some questions.
What about the inducibility of this repair? Is it inducible by exposure?
Yes, we actually in the manuscript that we published in Mutation Research on using DNA repair as a biomonitoring tool, we actually looked at inducibility. And it appears, and we also found in vitro, that nucleotide excision repair is very well inducible. But the evidence for an inducible base excision repair is less strong. I do think that base excision repair may be to some degree inducible, but the nucleotide excision repair can be induced much better. There is another problem, is that these two systems cannot be seen as two separate entities. Because nucleotide excision repair can also repair lesions that are usually recognized by base excision repair, and so there is some overlap in substrate between the two systems. So if you would induce nucleotide excision repair, you would also see a repair in 8-oxo-DG. It's not as clear. But according to the literature that we describe in our review, the nucleotide excision repair is much more inducible than base excision repair.
That is very interesting because it's quite likely that basic excision repair is constitutive because there's always oxidative damage going on in the body, whereas we're not always exposed to UV or benzpyrene, so nucleotide excision repair is more likely to be inducible.
Yeah, but there's also an idea that nucleotide excision repair might be something like a backup system for excessive oxidative stress-related damage, and then it would be logical to induce it, right? I see a question with no studies relating base excision repair Comet results with OGG1 sensitive sites. No, we don't have those data in the database. The only data we could relate the DNA repair assay to was to FPG Comet assay. So we don't have data on the OGG1 sensitive sites. But I think very interesting question, and maybe something we have to look into.
You mentioned briefly the challenge assay compared with the in vitro assay. And one of the things we haven't done, unfortunately, is to see whether they correlate, whether the challenge assay gives the same results for an individual as the in vitro assay.
No, we did not do that in Workgroup 5 or in hCOMET in general. But for the nucleotide excision repair, we actually did something similar when we developed the assay, at least at Maastricht University. Sabine, she actually compared the removal of BPDE-related DNA adduct, so measured by post-labeling, and related that to the nucleotide excision repair activity, and she found a very nice correlation between these two. For the base excision repair assay, we don't have such a correlation or comparison, but I think that's also something we could have a look at later on